Compare and contrast movement of molecules through a membrane. Within our cell membrane, we've got a phospholipid bilayer. The phospholipid is an amphipathic molecule which has a phosphate-containing heads with a charged region, which is going to make it hydrophilic. We've got fatty acid tails. The fatty acid tails are going to be lots of non-polar covalent bonds, which are going to contribute to a hydrophobic barrier, so that materials that are suspended with water either outside of the cell or inside of the cell are going to have difficulty passing through this hydrophobic barrier within our cell membrane. So if we need to move materials across the membrane, we need to be able to determine what are their characteristics and are they capable of passing through the membrane on their own, or are they going to require some type of path through the membrane. Now, the first molecule we can take a look at is going to be water. Water is going to be a very small molecule when it's free, and it's going to be capable of kind of moving through the membrane on its own at a slow pace. Or we could have aquaporins, which are examples of proteins, transmembrane proteins that are embedded within the membrane that form a hydrophilic path or hydrophilic tunnel through the membrane for these water molecules to pass through it. And these are going to be very important when we study a water movement within the kidneys. How do gases move across the membrane? Gases like oxygen up here to the top and carbon dioxide down here at the bottom are going to be nonpolar molecules. They're going to be able to be dissolved in water at a low amount, carbon dioxide a little bit more than oxygen, but they're going to be able to be within the water, and because they are nonpolar and they're very, very small, they're going to be able to diffuse directly through our phospholipid bilayer. And so they're not going to be blocked by the hydrophobic characteristics within the membrane. They're going to be able to pass through the membrane directly. How do ions move across the membrane? So ions are going to be charged particles. So we've got an example here with sodium with a positive charge, chlorine with a negative charge. Individually, ions are going to be very small. But what's going to happen in solution is that our ions are going to interact with water molecules. And so that positive charge of a sodium here, because it's given off an electron, is going to be attracted to the partial negative of water molecules. So the oxygen of water molecules, the oxygen in water molecules is forming a polar covalent bond with hydrogen, so it's holding the electrons closer to it, so it's got more of a negative charge. And so what we're going to have, instead of free ions in solution, we are going to have the ion with its charge surrounded by a hydration shell. And that hydration shell is going to make this molecule, or make this ion, too large to pass through the membrane on its own. Similarly, chlorine with its negative charge, it's gained an entire electron, and it is going to be attracted to the partial positive charges of our hydrogen water molecules up here. So again, chlorine with its negative charge is going to have a hydration shell around the outside. And so it's not going to be able to pass through that hydrophobic barrier within the phospholipid bilayer all on its own. It either needs to have an ion channel, like a tunnel through the membrane here, or some type of pump, which is going to be basically moving the materials through a protein complex so that these materials aren't exposed to the hydrophobic characteristics within the fatty acid tails at the core of our membrane. And so ions are going to have to be able to pass through either a protein ion channel or some type of protein carrier molecule or pump like we've got illustrated here. How do polar molecules move across the membrane? Polar molecules, like ions, are going to have a distribution of charges, and so polar molecules often are going to have a partially positive region, a partially negative region. Those partially positive and partially negative regions are going to attract water molecules to interact with them. They, in essence, are going to end up with a hydration shell around the outside. And so those molecules, polar molecules, are not going to be able to move through the membrane unless we've got a protein that is facilitating that. Generally, when we're talking about polar molecules, we're talking about larger molecules, and so we're going to have something more complicated than an ion channel. We're going to have some type of carrier molecule that is going to be involved with physically moving the materials either down their concentration gradient gradient or in many cases against their concentration gradient. And so we're pumping them across the membrane using some type of protein complex and the shape change within that protein complex to move the materials across the membrane. How do nonpolar molecules move across the membrane? So nonpolar molecules are going to want to interact with the fatty acid tails, and these nonpolar molecules are going to be hydrophobic. They're not going to interact well with water, so often what we can see, in this case kind of illustrated like a steroid hormone like testosterone or estrogen, out here within the extracellular space, we need to have some type of carrier molecule or molecule that's going to assist in transporting that. It is going to bind to our steroid hormone nonpolar, built from cholesterol. The carrier molecule is going to allow it to be able to transport it throughout the fluids of our body. When it gets close to the cell, that transport molecule, that carrier molecule, is going to release it. That can then diffuse easily across our phospholipid bilayer, but then it's going to come to the aqueous region inside of our cell. And so generally there's going to be another carrier molecule, another transport molecule inside of the cell that's going to bind to our hydrophobic steroid molecule, steroid hormone like estrogen or testosterone. It's going to bind to that and then move it through the cytoplasm, in this case move it into the nucleus, where it's going to have the effect of altering gene transcription, so changing the proteins that are turned on within this cell. So nonpolar molecules can move across the membrane relatively easily, but they need carrier molecules to carry them in the aqueous environment outside of the cell and then the aqueous environment in the cytoplasm.